Hello everyone and welcome. I'm Dr. S.K. Singh, holding a PhD, and I'm excited to be here with you today. Today's presentation is all about microbial contamination control in sterile cleanrooms. We'll explore key strategies and best practices to maintain a contaminant-free environment. So, let's get started and dive into this important topic together. Welcome to this part of the presentation. Today, I want to emphasize that sterile products must be completely free from viable microorganisms. This is crucial to ensure patient safety. Contamination risks are present at every stage of the manufacturing process, from raw materials to final packaging. It's important to recognize that contamination can occur at any point if we're not vigilant. We've seen a significant regulatory shift in how we approach this challenge. Instead of relying solely on end product testing, the focus is now on proactive prevention. This means implementing controls throughout the entire manufacturing lifecycle to catch issues early on. The ultimate goal here is to ensure both patient safety and sterility assurance. We want to make sure that every product reaching the patient is safe and sterile. And as highlighted in Annex 1, the focus isn't just on final testing. It emphasizes life cycle contamination control, meaning we need to manage and monitor contamination risks throughout the entire process, not just at the end. This shift really underscores the importance of a comprehensive, proactive approach to contamination control, aligning with the overall theme of strengthening our quality systems. Today, we're focusing on the regulatory framework that underpins our quality standards. One of the key global standards we need to be aware of is the EU GMP Annex 1. This annex was updated and became effective in August 2023, reflecting the latest expectations for manufacturing practices across the industry. It's important because it sets the benchmark for sterile production and contamination control worldwide. Annex 1 aligns closely with two important international guidelines, ICH-Q9, which covers quality risk management, and ICH-Q10, which describes the pharmaceutical quality system. These standards help us integrate risk-based thinking and a robust quality system into our operations, ensuring consistency and safety. One of the most critical requirements in Annex 1 is the Mandatory Contamination Control Strategy, or CCS. This isn't just a suggestion, it's a core part of compliance. The CCS involves a comprehensive approach to identifying, assessing, and controlling sources of contamination throughout the manufacturing process. It's about proactively managing risks to ensure that contamination is minimized, which is essential in sterile environments. Connecting this to our overall theme, understanding and implementing these regulatory standards and the CCS is fundamental. They guide us in building a culture of quality and safety, which ultimately protects patients and enhances our reputation in the industry. Let's talk about what microbial contamination actually is. Essentially, it refers to the presence of microorganisms like bacteria, fungi, and spores in our environment. These tiny entities can be found almost everywhere, on personnel, in the air we breathe, on surfaces, and even on the equipment we use daily. It's important to understand that these microbes aren't just harmless visitors. Some can produce harmful substances called endotoxins, and they can form complex structures known as biofilms, which are particularly challenging to eliminate. These biofilms can protect the microbes and make contamination much harder to control. So, controlling microbial contamination is a key part of maintaining a safe and sterile environment, especially in sensitive settings like healthcare or manufacturing. Recognizing where these microbes come from and how they can persist helps us develop better strategies to prevent their spread and protect both our health and our processes. Let's take a closer look at cleanroom classification. Starting with Grade A, which is the critical zone, specifically designated for aseptic operations. This is where the most stringent controls are in place to prevent contamination during processes like filling. Next, we have Grade B, which acts as the background environment for Grade A. It provides a controlled environment that supports the Grade A area without compromising its cleanliness. Then, there are grades C and D, which are considered less critical areas. These zones are still controlled environments, but with less stringent requirements compared to grade A and B. It's important to note that this classification system is aligned with ISO 14644-1, ensuring standardization across the industry. For example, in a manufacturing setting, the filling zone would be classified as grade A, while preparation areas might be grade C or D. Understanding these classifications helps us ensure the right level of cleanliness and control at each stage of production. Let's take a closer look at the key regulatory concept of CCS, or Contamination Control Strategy. This is a facility-wide, documented approach that helps us systematically control contamination. It starts with identifying the sources of contamination within the facility. Understanding where they originate is crucial. Next, we pinpoint the critical control points, or CCPs, which are specific stages or areas where contamination risk is highest and where we need to implement controls. This ensures that we're focusing our efforts effectively. The CCS then integrates several key elements: the facility design, personnel practices, and cleaning and monitoring procedures. All of these work together to create a comprehensive, cohesive strategy that minimizes contamination risks throughout the manufacturing process. It's important to remember that this approach isn't just good practice. It's a mandatory requirement outlined in Annex 1. By implementing a robust CCS, we're not only complying with regulations, but also ensuring the safety and quality of our products. This strategy truly underpins our overall quality management approach and helps us maintain the highest standards in our operations. Now, let's take a closer look at the sources of microbial contamination. First and foremost, personnel are the primary source. This means that the people working in the environment can unintentionally introduce microbes through skin, hair, or clothing. That's why proper hygiene and gowning procedures are so critical. Next, airborne contamination plays a significant role. Microbes can be carried through the air, especially if airflow isn't properly controlled or filtered. This is why cleanroom design and air filtration systems are essential to minimize these risks. Surfaces and equipment are also common sources. Microbes can settle on surfaces or hide in hard-to-clean areas, so regular cleaning and sanitization are vital to prevent microbial growth. Utilities such as water and HVAC systems are other key sources. Contaminated water or poorly maintained HVAC systems can spread microbes throughout the facility, so monitoring and maintenance are crucial. Finally, raw materials themselves can introduce microbes. Ensuring that raw materials are properly tested and handled helps prevent contamination from the very start. All these sources highlight how contamination can come from multiple points, reinforcing the importance of comprehensive control measures across the entire process. Let's now discuss personnel control. First, it's essential that everyone involved has the proper training and qualifications. This ensures they understand the critical procedures and maintain high standards throughout the process. Next, gowning systems are a key part of this control. Wearing sterile garments correctly helps prevent contamination. Behavior control is equally important. We need to move slowly and deliberately to avoid accidents or mistakes. Also, unnecessary talking should be minimized to prevent distractions and maintain focus. All these measures work together to uphold the integrity of the sterile environment and ensure safety and quality in our operations. Let's talk about environmental monitoring, or EM, which is a crucial part of maintaining a sterile environment. There are two main types we focus on: viable monitoring, which tracks microbial presence, and non-viable monitoring, which looks at particles. The key requirement here is continuous monitoring, especially in grade A areas, where maintaining the highest standards is essential. In grade A environments, the expectation is 0 CFU, meaning no colony-forming units, or in other words, no microbial growth should be detected. This is critical because even a small contamination can compromise product safety. If we do detect any colonies during monitoring, that immediately triggers an investigation. It's a clear signal that something has gone wrong, and we need to identify the source and take corrective actions right away. So, continuous monitoring helps us catch issues early, maintain compliance, and ensure the integrity of our products. Let's talk about cleaning and disinfection. It's crucial to have validated cleaning procedures in place to ensure effectiveness. This means following established protocols that are proven to work consistently. Now, an important aspect is the rotation of disinfectants. Using different types, such as alcohol and sporicidal agents, helps prevent the development of resistant spores. If we rely on the same disinfectant all the time, we risk creating resistant microorganisms that are harder to eliminate. When choosing disinfectants, we need to focus on two key factors: contact time and coverage. Contact time is how long the disinfectant remains on the surface to effectively kill pathogens. Coverage ensures that all surfaces are thoroughly disinfected without gaps that could harbor microbes. In summary, validated procedures combined with proper disinfectant rotation, paying attention to contact time and coverage, are vital for effective cleaning and disinfection. This approach helps us maintain a safer environment and prevent resistant organisms from taking hold. Let's take a closer look at facility and HVAC controls. These are crucial for maintaining a sterile environment. HEPA filtration, for example, is used to trap very small particles, ensuring the air remains clean. Laminar airflow, which is unidirectional, helps prevent turbulence that could stir up contaminants. Pressure differentials also play a key role. They keep clean areas at a higher pressure than surrounding zones, so contaminants can't flow in. When we qualify a clean room, we check airflow patterns, filter integrity, and microbial presence to ensure everything is functioning properly. For instance, airflow turbulence can cause contamination, especially during aseptic filling, which is why precise control is so important. Now, moving on to advanced technologies. Isolators and RABS, or restricted access barrier systems, are designed to create a physical barrier between the product and the environment, significantly reducing contamination risk. Rapid microbiological methods, or RMM, allow us to get microbial test results faster, speeding up quality assessments. Automation is also a key player here. Reducing human intervention helps minimize the chance of contamination. For example, using an isolator can decrease human contamination risk by over 90%. Overall, these innovations are vital for ensuring product safety and maintaining high standards in our sterile manufacturing processes. Let's take a closer look at the risk-based approach, or QRM, which is at the core of our quality risk management process. The first step is to identify the risks that could potentially impact our processes or product quality. Once we've pinpointed these risks, we move on to assessing their significance and likelihood. This helps us prioritize which risks need more attention. Next, we implement controls to mitigate or eliminate these risks, ensuring our processes stay within acceptable limits. Finally, we review the controls and the overall risk management process to make sure everything remains effective and up to date. Now, to support this approach, we use specific tools like FMEA, or failure mode and effects analysis, which helps us systematically evaluate potential failure points and their impacts. Another valuable tool is HACCP, which is particularly useful for identifying hazards and establishing critical control points, especially in food and pharmaceutical manufacturing. It's important to note that Annex 1 mandates all processes follow these QRM principles. This ensures a consistent, science-based approach to managing risks across the board. For example, let's consider the risk assessment of an aseptic filling line. We would start by identifying potential risks, such as contamination or equipment failure. Then, we'd assess how likely these risks are and what impact they could have. Based on that assessment, we apply controls, like sterilization procedures or equipment maintenance schedules, and regularly review these measures to ensure they remain effective. This structured approach helps us maintain product quality and patient safety at every step. In this case study, we're looking at a scenario involving microbial contamination during vial filling, which is a critical step in our manufacturing process. The root cause turned out to be operator glove contamination, highlighting how even small lapses in aseptic technique can have significant impacts. As a result, we saw batch rejection and even regulatory observations, which underscore the importance of strict contamination control. To address these issues, we implemented several corrective actions. We enhanced operator training to reinforce proper aseptic practices, increased the frequency of environmental monitoring to catch potential issues early, and updated our contamination control strategies to prevent recurrence. This example really emphasizes how identifying the root cause and taking targeted corrective actions are essential for maintaining product quality and compliance. It also ties back to our overall focus on continuous improvement and rigorous contamination control in our manufacturing processes. Let's wrap up with the conclusion. Microbial control is really about being proactive rather than reactive. That means anticipating potential issues before they happen and taking steps to prevent contamination. CCS, or contamination control strategies, serve as the backbone of compliance. They help us stay aligned with regulations and ensure product safety. When it comes to key success factors, a strong quality risk management system is essential. It guides our decision-making and helps us identify and mitigate risks effectively. Continuous monitoring is also critical. Keeping a close eye on processes allows us to catch issues early and maintain control. And of course, personnel discipline is fundamental. Well-trained, diligent staff are our frontline defense in microbial control. Remember, failure in any of these areas can lead to serious consequences: recalls, regulatory actions, and most importantly, risk to patient safety. So, maintaining a proactive approach and focusing on these key factors is vital for success in our microbial control efforts. For further support, please feel free to contact us. We're here to help you with any questions or additional information you might need. Thank you so much for your time and attention today. We truly appreciate your interest and engagement. Remember, we're committed to supporting you beyond this presentation, so don't hesitate to reach out. Once again, thank you.